SpongeBob has ate a bad Krabby Patty, and now a mutation is happening to his color. For us to see the mutation, we have to go back to where transcription and translation occurred. Transcription is DNA to RNA. Translation is RNA to protein. The first step SpongeBob's color gene is going to have to go through is transcription. In transcription, RNA polymerase unzips the double helix DNA. This process is known as initiation, showed by the black box on the left-hand side of the RNA polymerase. The next process is the elongation, where RNA polymerase makes a complementary RNA strand from the antisense strand shown on the diagram as the template strand. Elongation is shown by the red RNA, where the RNA-DNA hybrid region is being extended out and is having the RNA come out and the DNA wrap around itself again. The final step is termination, where RNA polymerase meets a terminator, represented by the black box on the right-hand side of the RNA polymerase, and leaves a completed RNA single strand left over. But the RNA isn't completely finished after transcription. First, it's got to be edited, and the free mRNA is going to have the introns and exons all together. The introns don't do anything for the protein later on in translation. But before we can remove the introns, we got to add a cap and a poly A tail onto the front and back end of the free mRNA. Then you can remove the introns by cutting them out and splice together all the exons to have a completed mRNA. Now that the RNA is done, it will leave the nucleus on a messenger RNA and enter the cytoplasm. The next step is for the transfer RNA to bind its anticodons with the messenger RNA codon. They do this through the ribosome. The transfer RNA and the messenger RNA will start at the codon AUG and continue to bind until the ribosome reaches the encodon UGA. The transfer RNA will enter the ribosome through the amino acid site and then grow the protein chain along the peptide site and exit versus the acid site. As the codons and anticodons meet, they make proteins, which are linked together in peptide bonds. Once the protein is done, it is released. Now that we know how SpongeBob has had his DNA transcribe itself into RNA and translate itself into a protein, we can now see where his mutation occurs from eating the Krabby Patty. The specific mutation that SpongeBob faced was a point mutation called substitution. In this, the C from frame 1 on the top is changed out with the T from frame 2 on the bottom. This is also known as changing cytosine with thymine. In result to those things changing, this will change the amino acid statement and lead to SpongeBob turning green.